Thank you for the introduction, and thank you again for the opportunity to obviously receive this award, but also present some of our work. So my lab really is focused on two things. One is developing new laser and optical-based techniques for imaging the body, and then the other is using those techniques to study the brain. And specifically, we're interested in the blood vessels in the brain and blood flow. So if you think about the brain for a minute, the vascular anatomy is really amazing. Of the 5 liters or so of blood in the body, in the human body, that 5 liters of liquid is distributed throughout the entire body over thousands of kilometers of blood vessels. And in the brain, there's actually hundreds to thousands of kilometers of blood vessels just within the brain. Another way to think about that is to consider a meter of blood vessel. So take a meter of blood vessel, now coil that up, and in some parts of the brain, 1 meter of blood vessel is contained within 1 cubic millimeter of tissue. And so when you compress that 1 meter of blood vessel down to a volume that's 1 millimeter on a side, it still only occupies less than 3% of that volume. So, of course, that means the blood vessels have small diameters. But the reason for this highly complex network of blood vessels, specifically in the brain, is that the blood vessels are, of course, supplying oxygen and nutrients to the brain. And of the billions of neurons in the brain, each one is located no more than about 20 microns from the nearest blood vessel. So the neurons really need this highly complex network of the vasculature for their survival. Now, there are a number of neurological diseases and disorders that are highly dependent on this vasculature and are affected by this vasculature, stroke being the big one. So when someone has a stroke, a blood vessel is blocked or is bleeding, and that leads to the classic neurological effects of motor impairment, language impairment, and all the others. And the impact and severity of a stroke depends a lot, as you might imagine, on which blood vessel is blocked and where it's located. And most of the strokes that people see clinically are blockages of large vessels. But what happens when one of those tiny blood vessels is blocked? Probably nothing if it's just one single capillary. But how many vessels need to be blocked or occluded before you start to see a noticeable effect? There's also several new hypotheses about the vascular basis for other neurological disorders. Things like even like Alzheimer's disease may have a vascular component to it. So what we're interested in doing is developing techniques to study the blood flow and the microvasculature, both clinically and also look at some of the more basic science side of things where we can start to address the question of what happens when you occlude one tiny blood vessel in this sea of vasculature. So what I'll talk about today are really those two things. One is the clinical application of a very simple technique that we've developed to image blood flow, and the other is a preclinical application of trying to image this dense vascular network and then interrogate the function of that network. So the first, one of the techniques we've developed and been using for a number of years now is very, very simple. And it's based on a well-known physical phenomena of laser speckle. And you've probably all seen laser speckle before, whether you've noticed it or not. And it's if you take even just a laser pointer and you shine it on your finger or a wall, you may have noticed that there's a twinkling. That twinkling is laser speckle. It's a random interference pattern. And the reason that it's twinkling is that that random interference pattern is fluctuating in time. It's called dynamic light scattering. And essentially what happens is that the dynamics of that twinkling, the intensity fluctuations, contain some information about the motion. So if you imagine a simple experiment where you just take an ordinary laser, expand the beam and illuminate an area of tissue, and then just look at it with a simple camera, you see something like this. So you see that there's this grainy pattern. It sort of looks like snow on your TV, but it's fluctuating. Okay, this is the reflectance of a simple laser from the surface of the brain. This is in a rat model where we're looking directly at the surface of the brain. The skull has been thinned essentially to transparency. And the fluctuations arise because something's moving. And in this case, it's the blood flow. So what we can do is try to quantify that. So here's another image. This is a single speckle image. And so what happens is that the fluctuations, that twinkling, gets integrated over the exposure time of the camera. And the net result is that we end up with spatial variations in the smoothness of this speckle pattern. And so the two squares there actually are highlighting two regions of this seemingly noisy image that have different degrees of blurring of this speckle pattern. It's maybe a little hard to see here, but one of them is a region which happens to be over a blood vessel where the speckle pattern is really blurred out and it's relatively smooth, and the other region is one where you have a lot of variation in the speckle pattern. So what we do is we simply calculate the spatial variance within this image to quantify the spatial smoothness of this speckle pattern. And if you do that in a local region and slide that moving window across the entire image, what you end up with is this. So the one on the right is calculated from the one on the left. So all of that information is contained in that seemingly useless image on the left. And we can, what the colors on the image on the right are showing or highlighting is not just where the blood vessels are located. It's actually a map that's related to how fast things are moving. So what we can calculate is blood flow, essentially, or something closely related to blood flow from these very simple images. And one of the advantages of this approach is that there's no dyes involved. All we've done is illuminate the area with a low-power laser and use a camera and then a computer. So we can acquire these images at anywhere from tens to hundreds of images per second, which allows us to visualize blood flow in real time. And so what I'll show here is a movie where we can visualize the dynamics of the blood flow during the formation of a clot, a very small clot. And this is in a mouse now, where we're looking at the surface of the brain, and the colors are showing where blood flow is highest. So if we can play the movie, you see some just normal baseline fluctuations, and then we actually introduce a second laser, which will actually initiate a clotting cascade to block the blood flow. So can we play the movie? Okay, there we go. So these are just normal fluctuations, and then this is sped up. The whole time course here is over several minutes. And around now, we turn on a green laser that actually activates this clotting cascade. And what we see is as the clot forms, the blood flow is blocked from certain branches. So there, that one's blocked, and then that one. So that branching vessel there is now occluded. And the clot is actually breaking away and reforming. So there it just broke away, and there you can see a portion of that vessel is actually, the clot is partially occluding that vessel, and we can see the blood flow creeping around the edge of that clot. And that blood vessel is about less than 50 microns So this is a very, very simple technique that allows us to visualize this clot formation. So really, to sort of drive home the point that this technique is so simple, I had a team of undergraduates implement this laser speckle imaging with a laser pointer and a webcam and actually do a stroke study with that instrument. And so you can see we took apart a webcam, and so the webcam is the green board there is the inside of the webcam with the shell removed, and we used a couple of plastic lenses that are used in cell phones, and then to the right of that is a laser pointer. And then the whole thing was used, and the image on the right is an example of highlighting the blood flow in a branching vessel in a mouse that is highlighting the flow through that vessel that had been occluded. Now, here's an example of how we can actually use this to study the chronic effects of blood flow and blood flow restoration after stroke. So the image on the left is under normal conditions, and this is in a mouse, and this is over only really about a few millimeters field of view. And then the second image is immediately after we induce a stroke. You see this very large blood flow deficit. The blue, of course, is blood flow that's essentially zero. But then over time, in this case over about a week after the stroke, we see a partial reperfusion and restoration of blood flow. And so this technique can now be used in conjunction with rehabilitative strategies even in rodents. So we can then relate the effectiveness of rehabilitation, post-stroke rehabilitation, with the actual underlying blood flow restoration and deficits. Now, one of the big advantages, like I mentioned, of this technique is that there's no dyes involved. It's really just low power light and a camera. So what this enables us to do is to do this in humans during neurosurgery. Now, one of the limitations of the technique is that it does not work through the intact skull. However, we can do it during neurosurgery where in many cases the skull has been removed. So what we've done is work with some neurosurgery collaborators at St. David's Hospital in Austin to incorporate the instrumentation into the surgical operating microscope, and which really involves just incorporating a laser diode and camera into the operating microscope. And so here's an example of the use of this instrument. One of the nice things is that since we're adding this on to the existing surgical operating microscope, there's no need for the surgeon to introduce a new instrument into the surgical field. Our instrumentation fits within the normal sterile draping of the microscope, and so the surgeon can literally visualize blood flow at the flip of a switch simply by turning the laser on and off. So unlike many other intraoperative monitoring techniques, there's no disruptions to the normal surgery. And so here's a side-by-side comparison of what the surgeon sees through the regular reflectance image of the operating microscope and the blood flow map that we acquire simultaneously. And so the image on the left, of course, is you can clearly see all the blood vessels, and this now is over a much larger field of view since this is human brain. This is about 4 to 6 centimeters on the side here. And what the blood flow image shows is the very small blood vessel coming up from the bottom actually has much higher blood flow than some of the surrounding vessels. And that vein in the top left region of the image actually has lower perfusion than some of the others. And that's probably not immediately obvious just from the reflectance image on the left. And here's another example of a blood flow image after a portion of the cortex had been cauterized with electrocautery as part of the normal surgical procedure. And so one of the consequences of this sort of electrocautery is that you end up with a region of tissue with significantly reduced blood flow, essentially an ischemic region. Now, there may not be much the surgeon can do about this, but at least this technique now can give the surgeon immediate feedback about the effects of the normal course of the surgery. Now, we're in addition to this just real-time monitoring of blood flow, we're also interested in the microscopic structure, this highly complex network of blood vessels that I mentioned before. So if we consider a speckle image like this is now in a mouse, and this is only about 4 millimeter field of view here. If you look at what a small subregion of that image, the contributions to the blood flow that we measure actually come from that. So within that small subsquare, what's shown on the right is another technique that we use, which is multiphoton fluorescence microscopy, which is a fairly well-established technique now for three-dimensional fluorescence imaging of the microvasculature. And so this now really reveals the microscopic structure of what it is we measure, even intraoperatively. And so what this technique allows us to do is get these highly detailed maps of single blood vessels. And so if we can play the movie on the right, this is sort of a fly-through of the images that we get layer by layer as we go from the surface down to only 0.5 millimeter deep within the mouse cortex. So what we're seeing here is just the blood vessels. And you see all of that dense vasculature that I mentioned before. So we can now take this image stack and combine it together. And this is what it actually looks like in 3 dimensions. And using image processing techniques, we can actually, what we call vectorize the vasculature, meaning segment the vasculature and identify every single vessel segment within this region that's shown in the inset. Each of those colored segments is one blood vessel segment. And along that segment, we know the diameter of the vessel and the centerline vector of the vessel and the branch points between adjacent blood vessels. So it's really a true 3-dimensional vectorized map of the vasculature. This is very computationally intensive, but it then allows us to predict, both through experiments but also through computational modeling, the impacts of what happens when one of these small vessel segments or a number of vessel segments gets occluded. And that's shown here. So the image on the left is one of these vectorized vasculatures, and then on the right is the colored overlay is showing us what it is we actually sample with the real-time speckle imaging. And so finally, I'll just mention very briefly, a new avenue of our research that we're developing is actually building our own customized lasers, ultrafast lasers. These are femtosecond lasers to do this highly detailed 3D imaging. I mentioned on the previous slide that we have these highly detailed maps of the microvasculature only down to about 0.5 millimeter in tissue, and these are in mice. So we've actually gotten into building our own ultrafast lasers in an effort to push the depth that we can image with micron resolution down beyond 1 millimeter deep. And so we've recently actually built a diamond laser made from a synthetic diamond crystal. This is an ultrafast laser. And what that allows us to do is, if we can play one last movie on the right there, is now do the same sort of layer by layer imaging all the way down through a full millimeter of tissue. Again, seeing all of the single blood vessels, and it may be hard to see, but you can actually see single red blood cells passing through each of the vessels as well. So In conclusion, I hope I've convinced you that using some very simple optical techniques, we can actually gain a lot of information about blood flow and perfusion in the brain, and then also start to address some larger questions about the role of the structural aspects of the microvasculature in blood flow and stroke. And finally, I'd just like to acknowledge all the many students and postdocs that did all the work. Thank you. Okay.
The temperature, is there any effects on temperature of the brain when you do that, particularly when you were looking at human brain?
It's a good question. Yeah, so the question was, are there any effects on the temperature in the brain as we do this? It's an excellent question and something we've had to address with our IRB boards, et cetera. And it turns out, no, there's no, the laser illumination has essentially no effect on the temperature of the brain. And actually the intensity of the laser illumination that we introduce to the surface of the exposed brain is only a fraction of the amount of light that's introduced with the normal white light illumination from the surgical microscope. And it's at a longer wavelength. And so no, there's no heating.
Related question. My understanding is a typical treatment for a stroke is to lower the temperature as quickly as possible for the patient that has a stroke. How does this impact what you are doing?
Yeah, so the lowering of the temperature is in effort to basically slow down everything, metabolism, et cetera. And so it's really buying time, because when you have an occlusion or a bleed in the brain, the neurons instantly have a reduction in the oxygen supply. And so if you can reduce their need for oxygen, then you can prolong the time before which they die. Now, in what we're doing, we don't do a lot of imaging of stroke intraoperatively, because most people with a stroke don't have the full craniotomy type surgeries. But what our animal studies allow us to do is to actually look at both the dynamics of the blood flow, and using other techniques, we can actually assess the viability of the neurons, and then actually look at the relationship between the neural activity and the perfusion. But we're not looking specifically at cooling. Yeah.
Maybe one more question. The laser speckle technique be extended to airflow instead of blood flow to the lungs or the lung capacity?
So the question was, can the laser speckle technique be extended to other areas like airflow? So I think I mentioned briefly that laser speckle is actually a one component or one implementation of a much larger type of problem of dynamic light scattering techniques. But yes, in theory, laser speckle could be used to measure any type of motion, including air motion, as long as there's some scattering from that, which I would expect there would be. Yeah.
In the absence of a full craniotomy, can you use fiber optics to look at a small incision for drilling into the skull?
Yes, so that's an excellent question. And like I said, one of the limitations of the way we're implementing it right now is that you do have to remove the skull. And we have talked with surgeons about implementing this through not just one fiber, but actually through a fiber bundle, an imaging bundle, essentially an endoscope, where you can put a small burr hole in the skull and then introduce just that bundle and get a regional readout without the need for a full craniotomy. And that's actually something we're working on. So yes.
I think maybe you can come up afterwards.
Okay.
I've got the sign back there.
Thank you.